So this project was about the biodiversity of rodents and rodent-borne diseases in forest and urban green spaces of Europe, and it was led by a broad interdisciplinary European consortium. So, we are living in an era shaped by pandemics, and many of which are driven by zoonosis. And human activities, they play a major role in the zoonotic crisis because we are disrupting ecosystems and undermining the critical role that wildlife biodiversity plays in controlling the spread of the zoonotic pathogens. And small mammals are key reservoirs of these zoonotic agents, and in Western countries, forests and urban parks are main places where humans and wildlife come into contact, and they are hotspots for rodent-borne zoonotic risks. So with BioRodDis, we wanted to study the intricate relationships between small mammals, the microorganisms they carry, and their interactions with humans. So we aim to better understand how their diversity shapes the prevalence and circulation of rodent-borne zoonotic pathogens in European forests and urban parks. Specifically, we are focused on 3 objectives: describing small mammal assemblages and their pathogens, modeling how changes in small mammals' diversity influence the spread of these pathogens, and engaging with stakeholders to enhance strategies for preventing zoonosis and raising public awareness. So here are some key results from our project. So first, we've developed a comprehensive database on small mammals and the zoonotic pathogens they carry in both forests and urban parks across 34 sites in Western Central Europe. We've surveyed over 4,000 small mammals, representing 18 species, and we have detected 4 families of zoonotic viruses and 10 genera of zoonotic bacteria. We found some important contrast between the ecosystems of forest and urban parks. While both have a similar diversity of rodents, the species composition differs. In urban parks, the biggest zoonotic risks come from rats and wood mice, and we are seeing pathogen spread mainly through direct transmission. In contrast, in forests, the risk is posed by species like benguls and yellow-naked mice, with pathogens that are often vector-borne, mainly transmitted by ticks or fleas. And finally, we identified that environmental factors are key drivers in the distribution of these pathogens. Pathogens respond differently to these environmental factors like temperature or humidity or habitat type. So looking ahead, climate change is likely to significantly alter the distribution of rodent-borne pathogens, affecting risk patterns at local but also across Europe. So the third breakthrough I want to highlight is our modeling of the relationship between biodiversity and zoonotic diseases. In France, we found that small mammal biodiversity can actually help regulate the circulation of certain pathogens, and this is what we call the dilution effect. For example, we discovered that a higher diversity of small mammals can limit the prevalence of orthopox virus. However, it's not all clear-cut. In some cases, we found no effect or even a positive effect of small mammal diversity on the prevalence of other zoonotic diseases like leptospirosis. So this shows us that the relationship between biodiversity and human health is complex and context-dependent. So it means we cannot assume biodiversity always works the same way across all diseases. And monitoring small mammals and the pathogens they carry is crucial, especially in response to human impact and climate change. So here are 2 more breakthroughs with major implications for stakeholders. So on the left, you can see that we have modeled the impact of rodent managers on zoonotic risk, focusing on a specific case, the rats and cereal hantavirus in the city of Lyon in France. Our findings show that when you reduce rat population, it initially lowers the circulation of the virus. However, there is a catch. If rodent control methods are not species-specific, they can harm small mammal diversity in urban parks, and in turn, this could decrease the virus in the rat population above a certain threshold of rodent control. Second, we analyzed responses from questionnaires on public knowledge and perceptions of rodents and rodent-borne zoonosis. And one key takeaway, also not surprising, is that people who know about or think they know about rodent-transmitted diseases are more likely to take precautions against zoonosis. So this reinforces the idea that knowledge is a critical foundation for making prevention strategies effective. So in conclusion, one of the very naive but key lessons I've learned through the BioRodDis project, and what I believe has been crucial to its success, is the importance of taking time and energy to build trust with stakeholders. And we provided clear and accessible information, and we actively consult with them. So we were able to engage partners and lay the groundwork for an effective transdisciplinary collaboration. And this trust has not only helped us achieve our goals, but also has paved the way for new projects to emerge. Thank you.